Oryza genome, but I'm going to give that talk tomorrow. I've been asked to give a plenary lecture tomorrow, so my science talk is going to be given tomorrow. So this is the first time I've given this talk on the revised 2020. So I hope you'll bear with me, and I hope I don't upset a few people out there. There are a few general statements in there that some people may not take entirely correctly. So anyway, the purpose of this revised 2020 and how to solve the 9 billion people question is really to kind of lay out a framework for the next 25 years on how I think and how Chief and I think we should go, and it's basically for opening up a discussion on the topic. So, as was discussed early on this morning, the principle that kind of gets me going every day is really what we call the 9 billion people question. And that is, how do we grow enough food to feed the world in 40 years, in less than 40 years? Rice feeds half the world. It's the rice-dependent population that will double by 2050. And new rice varieties are needed with 2 to 3 times the yield, but ones that require less water, less fertilizer, less pesticides, and less land. And these are the so-called green supercrops that you'll hear about during the course of the meeting, and which Chief Ashong introduced today. Okay. In 2008, Chief and colleagues from China proposed what we call Rice 2020. And the main idea, the bottom line behind the Rice 2020 was really to determine the function of all genes in rice with the goal of developing a toolkit for high-throughput, knowledge-based rice breeding. So I'm going to go through these themes, not one by one, but just in general, and then move on to an idea on how to solve the 9 billion people question. So the first theme proposed the development of enabling tools and genetic resources for the international community of scientists to conduct functional genomics. And then it was in 3 main areas: insertion collections, full-length cDNA collections, and artificial microRNA collections. The Rice 2020, in terms of the insertion collections, Yui Singh gave a wonderful talk today about what's available and how we need more mutants, and clearly the generation of flanking tag sequences of all the insertion lines should be prioritized. And one thing I was thinking today is that if really money was no object, maybe it would make sense to simply just sequence the insertion mutations that the lines that are already there, and identify all of the variation that's available within these lines. I mean, that could be a simple and easy idea to rather than spend all the time in getting the flanking tag sequences. So it's clear that the generation of a complete set of genetically mapped FTSs is critically needed as a resource to be dedicated toward completing this objective. A critical problem with these lines are that many practical and logistical problems are associated with the development of such important mutant lines, like maintenance and storage of viable seed stocks, and the ability to share and distribute large knockout collections across international borders without restrictions. Quarantine restrictions, MTAs. So an ideal solution would be to essentially have all MTAs removed or lifted, and another idea would be that all mutant resources would be deposited in 5 to 10 locations around the world, and the collections would be grown out on an annual basis for collective phenotyping and tissue collection. This would permit rapid distribution and the phenotyping of the world collection of knockout mutants in a timely, efficient, and coordinated manner. The other part of Theme 1 was essentially full-length cDNA collections. The full-length cDNAs are an elegant type of collection to have, but now with the decreased cost in sequencing, it's now realistic to predict the rice community is capable of generating full-length quantitative cDNA expression data from every developmental stage, every tissue type, and cell type of a growing rice plant, from seedling to flower to seeds, under a plethora of normal and key biotic and abiotic conditions. Such experimental conditions in tissues would also permit the coordinated and in-depth explorations of rice proteomes, metabolomes, methylomes, and onomes. The third part of the Theme 1C was artificial microRNAs. And essentially, generating of artificial microarrays for all genes should be given a high priority, and it's important that one or two research groups would be given this assignment and that such resources deposited into the public domain. And in turn, these collections could be systematically transformed into rice to begin to knock out all genes and gene families. Now there were 4 themes, 2, 3, 4, and 7, that I think could be grouped into one general theme. The first was the assignment of biological functions to every annotated gene. That included systematic phenotyping and characterization of mutants, systematic characterization of gene families. Theme 3 proposed a systems-wide understanding of epigenomes, expression profiles, and regulatory networks with 3 aims. And Theme 4 proposed a global analysis of the proteome and protein interactions. And Theme 7 proposed the establishment of a toolkit for knowledge-based, high-throughput rice breeding. So the ideal solution is with the advent of glasshouse and field and airborne level high-tech phenotyping capabilities coupled with advanced computational resources such as iPlant, it's now conceivable that our community could interrogate the majority of all rice mutants, elite cultivars, and land races, and wild relatives of rice in a detail that's never been dreamed of 3 to 5 years ago. And such data sets would allow breeders to design the ideal rice plant and model plant for growth and yield under every condition to achieve maximum yield and quality and minimum environmental impact, the so-called green super rice. Theme 5 was the establishment of a comprehensive understanding of natural variation of O. sativa and its wild relatives. Ken McNally gave a nice talk today about the status of the Rice Diversity Project. I won't get into that. But one thing I wanted to mention, though, is that there's not only the consortium that Ken's involved in, but there are many other consortia around the world that are sequencing these lines. And I think it's important that not only do the data sets get into the public domain, but the biological resources used to generate these important data sets are made publicly available across international borders without restrictions. What I'd like to say about the wild relatives is that they contain essentially a virtually untapped reservoir of genes that could be used for agricultural improvement. And by 2014, there will be a complete set of reference genomes available for all diploid Oryza species and hopefully some of the polyploid species. And I'll talk more about that tomorrow. In terms of bioinformatics, there was a theme on data management and exchange and sharing of information. I think phenotyping at multiple levels of genotype and genotyping of the genus, as discussed, will yield unprecedented amounts of data for the world's most important food crop. And the rice agricultural community in general must position itself to be able to store, manage, and share and make use of such large data. and complex data sets to address grand challenge questions of our times to achieve the ultimate goal of creating a sustainable world food supply. So the Rice 2020 and the green super rice concepts are important initiatives and bold concepts that need to be taken seriously and embraced by the agricultural community. And in reality, our community has only about 25 years to solve the 9 billion people question in order to supply the farmers with the seed that's necessary to feed the world. This challenge is enormous, and we must act now at a scale never before seen if we are to achieve food security needs of our children and beyond. A critical problem that I see is that although enormous strides have been achieved at all levels in plant biology in recent years, there has been a major lack of coordination between countries in terms of conceiving a unified plan aimed at solving world hunger. One exception to that, and there are many others, but is the International Rice Genome Sequencing Project. And this occurred when 10 nations joined hands and brains to embark on the generation of the highest quality, publicly available reference genome sequence for any crop plant that was published in 2005. The funding agencies of these 10 nations wisely recognized the importance of generating a gold standard genome sequence for the world's most important food crop. And this sequence has had enormous impact on plant biology and breeding in general. Other examples which were discussed today would be the GRISP project, the Hybrid Rice Development Consortium, and Rice Diversity Project, and others. So even though the rice genome has been utilized extensively to understand many aspects of rice biology, a coordinated international effort to systematically understand its genome and accessions and cultivars and land races and wild relatives to be able to design green super rice varieties adapted to all growing conditions is almost completely lacking. In fact, rather than working as a community to help solve the 9 billion people question, rice scientists, in my mind, are competing with one another to, for example, clone the next gene or QTL, write the best paper, get the next grant, a process which is repeated over and over again worldwide. Competitive science is extremely important and critical in the discovery process, but given our timeline on the world stage, research on rice must enter a new and bold era of worldwide, coordinated basic, translational, and applied research if we are to solve the 9 billion people question in 25 years. So, the proposed solution of creating new green super rice varieties that not only meet caloric and nutritional needs of our society, but also protect our environment with less resources spent on production, we propose a concept called $9 billion to help solve the 9 billion people question. The proposed solution is relatively simple, but will require an in-depth discussions from various stakeholder groups to work out specific details. It could also serve as a model for other agricultural initiatives. The initial concept would be to raise $2 to $3 billion that would be used to establish 5 to 6 international green super rice science and technology centers. Half the money would come from philanthropic organizations, and half would come from federal funding agencies on a dollar-for-dollar matching of funds. The goals of the Science and Technology Centers, these green super crop Science and Technology Centers, will be to perform coordinated basic, translational, and applied research aimed at creating green super crops with an emphasis on rice. The STCs would house state-of-the-art research facilities and regional greenhouse facilities, and including regional greenhouse, field, and aerial phenotyping centers to phenotype regional accessions, as well as a common set of accessions and populations that would be used to obtain phenotype data across a multitude of environments. Such a center, Chief Ashong showed today, which is their RICE high-throughput automating phenotyping center. I was able to visit that in Wuhan this summer, and it's really an amazing greenhouse. And I think that rather than going just with the only system that's out there, such as Lemnitech, it's important to really think about what type of measurements you want to make, and if a commercial system out there is what you really want. This it seems very cost effective, and they're putting a lot of work into this, and I think that we can learn a lot from this the system that was developed in Wuhan, but there are definitely other approaches to take. The STCs would also serve to translate basic discoveries from single investigator labs worldwide into practical solutions. And that could be achieved by providing matching funding to competitively funded individual consortium labs around the world. For example, an example that I've been thinking about is that one could imagine the establishment of 6 international green supercrop science and technology centers, one each in Africa, India, South America, China, the European Union, and North America. And regional funding agencies would each provide about $250 million US dollars for a total of $1.5 billion US dollars. And this would be matched by philanthropic organizations for a total of 3. And the science and technology centers would be built and partially staffed within 2 years of raising the funds. If these funds could be secured by January 2015, STCs could be completed and staffed by 2017, and that would give us about 20 years to achieve our common goal. That's not a very long time. And the remaining $6 billion would be used to support 6 science and technology centers at an average of $46 million per year per science and technology center, and would come from a combination of national, international, and international funding agencies and philanthropic organizations. So what is $9 billion? $9 billion is not a funding scale that plant scientists are really used to thinking about. However, it's not a lot of money in the scheme of the world economy. That dollar figure equates to essentially $1 per individual per 9 billion people. And to put that in perspective, I live in Arizona, and we're involved in a lot of interplanetary exploration. And it cost $3 billion to send the Curiosity to Mars, which landed in the summer of 2012 as a NASA project. And one could argue that NASA's Mars Science Laboratory will help save our planet, but that's in the very, very long term. We only have 25 years to solve this problem, the 9 billion people question, and we must act now if we want future generations to reap the benefits of Curiosity and other future planetary explorations. Okay, this example that I've discussed here is an ideal one, and obviously many specifics need to be worked out, but regardless, the main point is that we want to emphasize is we must act now before it's too late. And in our opinion, an international coordinated effort is a new paradigm to create green supercrops to help solve the 9 billion people question. On the bright side, we as a highly coordinated group of plant scientists and federal and philanthropic organizations can accomplish an honorable and achievable goal in our lifetime and positively affect the world as we know it and our children's and their children's future as well. So this is my plan that I'm going to throw out to you guys for moving forward. If you all feel that this is a a good idea. We would appoint a 9 billion people question committee, hold a series of NBPQ meetings, achieve consensus, write a report, submit to various agencies, federal and philanthropic, raise the funds, make it happen, and then solve the 9 billion people question. So that's pretty simple, but I really think it's doable, and it's something that we really need to think about and talk about in a deep way. And hopefully we can talk about some of that today. I'd like to end my talk by just thanking my co-conspirator, which is Chief Ashang. We climbed Mount Tai this summer, where Confucius used to hang out, and we talked a lot about this project and some ideas of moving forward to help solve the 9 billion people question. And then I'll end that with a thanks for listening, and I'd be happy to open up the floor for discussions, questions, and comments. Thank you.
Is that on? Yeah. Thanks, Rod, for a very interesting and thought-provoking talk. I'm still thinking now. But the one thing that did occur to me is, to what extent are we capable of doing this fantastic phenotyping that would be needed? We need to build now, and do we know what we actually want to measure and how to measure it? I just throw that question up. You don't necessarily have to answer it, but I think it's a really serious point.
Typically people have just been, measuring yield, plant height, really simple things. And do we know now what we really need to measure? I would say the answer is no, but that would involve a large discussion and really get out there and like you're measuring vein width, things like that. So I mean, you can you can go as far as you want to go. But I think it's as the first speaker talked today, targeting various types of stresses, biotic, abiotic stresses, things like that. Those are types of phenotyping that we there's things that we have to go for now, and then there's things that we have to go for later. But I think we can imagine going all the way from the whole plant level down to the single cell level. And that's what I would like to do. Thank you.
All right. Any more? If not, I would like to invite people to join us in a panel discussion, and also whatever contribution you want to make to whatever described and proposed by Professor Wing. Thanks again, Rod. Well, I'm going to introduce our next speaker, Dr. Blake.